This is induction video 1 on animal cells. There is a student version of this PowerPoint that you can download off the Moodle VLE and print it off, and you can fill it in as we go through. If you don't have access to a printer and can't do that, but you could grab a pen and paper. You'll be able to do it then. Don't worry if you can't download it. You think, oh, I can't label up the cell. This will be covered in class. It's not the only time you're going to go through this, so don't worry too much. But you will need pen and paper because you're going to have some exam questions and things like that. In this video, we're covering the topic of the animal cell. In particular, we're going to aim to identify animal cell organelles on diagrams and describe the function of cell organelles on diagrams. So that's our learning objectives that hopefully we should have achieved by the end of this video. Okay, so cells. Hopefully you are aware from GCSE that all living organisms are made up of cells. There are two types of cell. You can either be a prokaryote or a prokaryotic cell. These are very simple, they're smaller, and you would know them as bacteria. So bacterial cells are prokaryotic cells. Or the other type of cell is a eukaryotic cell. These are more complex. They have more structures within them. As an animal, because humans are animals, we have eukaryotic cells. Also, eukaryotic cells are plant cells and fungal cells. You on your course will look at animal cells and at plant cells as examples of eukaryotic cells, and you will look at prokaryotic cells in terms of bacteria. But this video today is looking at these animal cells that we have within us, and you will have covered this at GCSE. So what I want to know from you now is I want you to have a go and draw and label an animal cell from memory. Don't look it up. Don't go onto Google or anything. Just see what, flick forward. Just want to know, what can you remember? Try and drag it out of your head. Draw it, label it, and add some functions if you can. So pause the video now to do that. Okay, so hopefully you've drawn something like this from GCSE. It's going to slightly vary depending on which spec you did. But there are certain structures that I would expect you to know at this point. I would expect you to know the nucleus, the largest organelle found within a cell. Often you may know, often get told that it's the brain of the cell. It's not the brain of the cell. Please don't write it's the brain of the cell. It controls cellular activities. It's where you find the DNA, and we'll look at it in a bit more detail. You should know the cell membrane. You probably call it cell membrane or the plasma membrane. If you want to call it cell membrane, that's fine. The correct term is plasma membrane. Sometimes it's called the plasma lemma. It's all the same thing. Now look at the spelling, and this is important. It's B-R-A-N-E. Not like the brain in your head, B-R-A-I-N. You will get penalised for spelling, so you need to be able to spell brain, as in membrane, correctly. Often I'm told the cell membrane or plasma membrane holds the cell together, which of course it does, but that's not going to get you a mark or any points from the examiner. So the function of the cell membrane is to control what comes in and out of the cell, and we'll look at how that happens. Then I would expect you also to be aware of the cytoplasm, jelly-like matrix found within a cell. Probably you know that it's where cellular activities occur. And then this is where things vary depending on which GCSE spec you did. You may have heard of ribosomes. I'm hoping that you have. Ribosomes being very small structures in the cell where protein synthesis occurs. And some of you may also know mitochondria. So mitochondria being the site of respiration. Remembering that respiration isn't breathing. Respiration is releasing energy from glucose. If you're talking about breathing in the lungs, it's ventilation. So try not to get those confused, respiration and ventilation. Now, all these structures that you find within this cell here are called organelles. Okay, and organelles literally means little organ, and you only find them in eukaryotic cells, and they've all got their own specific function. Okay, and that's what we're looking at. Now, some of you may have mistakenly drawn a cell wall on your diagram. I find that a lot of students at this point get confused between animal and plant cells. Plant cells have a cell wall. Animal cells do not. Animal cells don't need a cell wall because we've got a skeleton to hold us upright. Plant cells don't have that skeleton, and they rely on the cell wall in part to hold them upright. So remember, no cell wall on an animal. Okay, so this is what you've got from GCSE. I'm going to add to it a little bit now. This is what I need you to know at this level. And you'll see there are a number of other structures I've popped in there. So we're going to go around each one in turn, label up our diagram, and then talk about what they all do. So hopefully you managed to download the diagram off of the Moodle, the VLE, but here it is. This is a diagram taken from an electron microscope. Electron microscopes are very, very high-powered microscopes that have got really high magnification. and they've got a really good resolution. So magnification being making something bigger, resolution being clear. So you probably used a light microscope at school, and you maybe looked at, I don't know, cheek cell maybe, perhaps an onion cell, and you probably saw cheek cell. You would have seen your membrane. You might have seen a little nucleus. Maybe you drew it or whatever. But to see all those internal organelles within the animal cell, you need an electron microscope. They're very expensive, they're very big, and they require specific training, and most schools and even most colleges do not have access to them. So it will be the electron micrographs that you're using to identify organelles from. And when you come and when we start lessons, we actually have electron micrographs that you can use and identify from. But for now, we're using a diagram. So let's start labeling up. So the first structure I want to talk about is the nucleus. So we're going to label up first, and I'm going to look at each organelle in more detail. So this is our nucleus. Largest organelle in the cell, there it is, and we can see this sort of darker structure here. Okay, that is called the nucleolus, and what it is, it's a darker stained area within the nucleus. It's the site of sort of RNA synthesis. RNA, you may not have heard that. We'll cover it, don't worry. It's also where ribosomes are manufactured. And surrounding the nucleus is something called a nuclear envelope, so it's sort of all around the outside. Sometimes it's a nuclear membrane, and that membrane or envelope has teeny tiny pores or holes in it to allow the RNA to move in and out of the nucleus. And we'll look at that when we look at protein synthesis in a bit more detail, and why that has to happen. The next organelle on there that I think you should know about is the mitochondria. I always think it looks like it's got little teeth there, I don't know, but this is a double membrane here, and the inner membrane is folded in and out. That's why you get that kind of wavy line in the middle. Increases the surface area, but we'll look at that in more detail. Remember, site of respiration there. Then we've got the cytoplasm, jelly-like substance. All the cellular activities happen here, such as protein synthesis, glycolysis, all sorts of other things. Then we've got the plasma membrane, so the cell membrane that goes around the outside. As I said, it controls what enters and exits. Now, here we're going to talk about ribosomes, and on my diagram here, ribosomes are here, these little black dots, and they're attached to another structure here. And we're going to call them ATS ribosomes. And the reason they're ATS ribosomes is that the ribosomes come in two sizes, depending if it's a prokaryote or a eukaryotic cell. Eukaryotic cells are bigger, so they have bigger ribosomes, so they have ATS ribosomes. Prokaryotic cells are smaller, so they have smaller ribosomes, so they're 70S ribosomes. And the structure that we can see all these little ribosomes attached to is called the rough endoplasmic reticulum. And yes, the spelling does matter. Okay, you need to be able to spell it. And it's spelled as it sounds, I always think, endoplasmic reticulum. Often you'll see it called rough ER, but you need to know how to spell it. And it's rough because when you look at a diagram or a picture of it or an electron micrograph of it, it looks like it's got little blobbles all over it. And these are all the ribosomes stuck all over it, making it rough. But there's also this type here, which is called smooth endoplasmic reticulum, because it's got no ribosomes on it, so it's smooth. Now, I always think the smooth endoplasmic reticulum here looks very like this structure up here. Okay, if you look, they look very, very similar. Now, this up here is our Golgi apparatus, Golgi body, Golgi complex. Doesn't matter which you choose to use, they will correct. So Golgi apparatus, complex, or body, I'll use apparatus. And it looks very, very similar down here to the smooth. Apart from, if you look here, we can see we've got these tiny little bubbles just budding off it here and here. And that's how you can tell it's the Golgi apparatus and not the smooth endoplasmic reticulum. It has these little vesicles, they're called, budding off. And here's another example of a vesicle here that's travelled to the edge of the cell and it's going to disappear out the cell. Vesicles are like transport bubbles, I suppose, transport envelopes that carry molecules around the cell. And a type of a vesicle that we need to know about in particular is a lysosome down here. Now, they tend to make it easy. Oh gosh, lysosome, a vesicle, how am I going to know? They're going to make it straightforward for you. So though it's an exam question, they might point to one of these and say to you, what is that? Well, it would be a vesicle because it's budding off the Golgi, as opposed to that there, which is a lysosome. We know this is a vesicle because it's going towards the edge of the cell there. But lysosomes contain really strong digestive enzymes, and they're like kind of the rubbish collectors of the cell. They get rid of all worn out organelles and things like that. And then we have got the last one here are centrioles, these two structures here. You find centrioles in a structure called a centrosome, and they are very, very important in cell division. If you remember mitosis and meiosis, your cell division from GCSE, we'll cover it again, so don't worry if you don't. During that process, the cell puts out like a spindle across which the chromosomes arrange themselves, and it contracts and pulls them apart. Well, the centrioles are important in the spindle formation. Okay, so these are all the parts. So what we're going to do now is look at each one in turn, talk a bit about its structure, a bit about its function. As I said, there is a PowerPoint, there is a version of this you can download that will have that table on it, or you can just draw your own. Now, obviously, this is the picture that I've given you. In the exam or in class or in lessons, you won't necessarily get this picture. You might get one that looks like that, or you might get one that looks a bit like that, or like that. They're all examples of animal cells. You'd have to be identifiable organelles from all of them. So over the course of the 2 years before that you're with us, you will see many, many different diagrams of animal cells, and you would never get an exam question asking you to identify all organelles at once. It's always bits of the cell that you need to be able to identify. Okay, so here's the table. So we're going to go through each one in turn, a bit of description of what it looks like, and then a bit about its function. So what I suggest you do, if you've got the student version, you can just fill it in or make your own table up. So each one in turn I'll talk about. I suggest you then pause the video, fill in your table, and you can rewind it if you need to, repeat a bit, can't you, if you need to. Okay, so we're going to start with the plasma membrane and the cytoplasm. Okay, so plasma membrane around the outside of the cell, as we know. It controls what enters and exits the cell. It contains what is known as a phospholipid bilayer. Okay, now a phospholipid bilayer, bi meaning two, it's a double layer, and it's made of basically fat, a type of fat, and within that it's got little protein molecules studded all the way through, and you will look at that sort of plasma membrane or the phospholipid bilayer in more detail. But basically these proteins, they control what comes in and comes out via the processes of active transport or sort of diffusion and things like that. Hopefully active transport and diffusion are familiar to you. It does also protect the cell from its surroundings, but the main function of that plasma membrane or that phospholipid bilayer is to control sort of what comes in and goes out of the cell, so transport of materials in and out of the cell. Cytoplasm, not much to say about the cytoplasm really. It's a jelly-like substance that fills the cell. Site of many metabolic processes occur here. Protein synthesis is a good example of it. Okay, so that's the plasma membrane and the cytoplasm. Nucleus, large organelle, spherical, or more or less. It's where you'll find your DNA, and it's surrounded by a double membrane, this nuclear membrane here. It's a double membrane, and you can see it's got little holes in it, nuclear pores, allow molecules in and out, and we call sort of jelly-like substance in the middle nuclear plasm. Don't have to worry about nuclear plasm, you don't have to worry about sort of nuclear pore too much, but it's where you'll find the chromosomes. They condense up and become visible during cell division. And then you've got this darker stained area here, the nucleolus. It's basically the function of the nucleus to control the overall function of the cell. Okay, it does store genetic information, and those pores allow substances to enter and leave, sort of enter from the nucleus and go into cytoplasm and vice versa. That dark stained area here, the nucleolus, that's a region inside it that consists of proteins and something called rRNA that we'll talk about in more detail. But basically it's where ribosomes are made or synthesized. And that's why it appears a little bit darker. Okay, so it's a synthesis of ribosomes and RNA occur in that nucleolus there. And our overall function of the nucleus is to control cellular activities. Okay, and there's a store of genetic information. Golgi apparatus, Golgi body, Golgi complex. It has many names, but it's a stack of sort of flattened membrane sacs, for want of a better word. And what you can see is all these little vesicles budding off from it, constantly being released by the Golgi body all the time. And what it does, it modifies molecules such as proteins, and it packages them in these vesicles for transport elsewhere. It will make lysosomes as well. But its main role, well, it's not the main role, the main role that you need to remember is to modify molecules like proteins for transport and making the lysosomes. And remember, those lysosomes, they are basically a sac of digestive enzymes, and they digest older organelles and pathogens. And then you've got these vesicles. Well, the general sort of function of vesicle is a small membrane-bound sac that transports sort of molecules. So Golgi body is packaging and modifying proteins. Remember, it doesn't make proteins, it just packages them and modifies them. And we'll look at that in a bit more detail in lesson. So it packages them and it modifies them, and then it puts them in these vesicles to send them off either out of the cell or around the cell somewhere else. And it also makes these lysosomes that aren't shown on here, but lysosomes are those membrane-bound sac with those digestive enzymes to sort of digest old organelles and pathogens there. So that's the Golgi body apparatus, vesicles and lysosomes there. Mitochondria. It's said to be a rod-shaped organelle. Looks a bit like a tic tac, I think, myself. It's got this double membrane, this inner membrane and this outer membrane here. And you can see that inner membrane's folded in. And that what gives its appearance that when you see an electron micrograph, it looks like it's got something going on inside there. It's that folded inner membrane, increasing surface area, basically. And it's got like a matrix or like a jelly-like substance in the middle there. And these foldings we call cristae. Okay. It's where ATP is resynthesized during aerobic respiration, and in cells that require a lot of energy, maybe a muscle cell for contraction, or maybe it's a cell in your digestive system, you know, like one on the surface digestive system where it's producing lots of enzymes or absorbing lots of your molecules that you've digested. They're going to need a lot of energy to do that, so they're going to have loads and loads of mitochondria. Also, sometimes you don't always see them in this sort of nice sort of cut in half. Sometimes, if you think like a cucumber, if you hold a cucumber and then cut the top off and stare down at it, sometimes they do that to it. So they're not always shown in that sort of ovally shape. Sometimes they show them as round circles where they've cut them down the middle. So please be aware of that. I think actually there might be a little cheeky little exam question on that in the exam question pack for you to do. But yeah, so they do show them in different planes, they call it. Sometimes they cut them horizontally, sometimes they cut them vertically. But yeah, so site of aerobic respiration, they've got that folded inner membrane to increase surface area, double membrane, got that jelly-like matrix in the middle where loads of chemical reactions are occurring, cristae are the folds, mitochondria, site of aerobic respiration. Endoplasmic reticulum and ribosomes. So, two types of endoplasmic reticulum, smooth ER, rough ER. Rough ER is called rough because it's covered in ribosomes. Smooth ER is smooth because it hasn't got any. And you will see from this little diagram here that it is what is said to be continuous with the nuclear envelope. The nuclear envelope just basically continues on in terms of ER. Now, it's not always shown like that in diagrams and exam questions. Quite often, mostly is attached to the nuclear envelope, but not always, so don't be fooled. If it looks like if it looks like that with spots all over it, it's rough ER. And if it's not got spots over it, it's smooth ER. So, smooth ER here, this stuff here, this smooth without any ribosomes on it, it's like they're like long tubular membranes, and they kind of they're enclosed in like a fluid-filled space. So like if you think in here, like these are like long tubes, and in here they've got like a fluid. And basically what happens here, it's all about carbohydrates and lipids. They're synthesizing and storing carbs and transporting lipids around the cell. But basically the thing to remember at smooth ER, it's about lipid synthesis for smooth ER. So that's the main thing to take away. Smooth ER, lipid synthesis. Whereas the rough ER is all about proteins. So again, it's still a system of these like long tubular membranes here, like these are long sort of tubey structures, and they've also got the fluid-filled space in them. This time they are all about proteins, because they've got the little ribosomes on the outside that are folding and that are sort of producing these proteins, and then in the rough ER they're kind of processing them, storing them. Yeah, so the rough ER is all about proteins, and the smooth ER is really all about lipids. There is some carbs there as well, but it's mainly lipids or fats, as you probably call them. So rough ER is proteins, smooth ER is lipids. Okay. And then ribosomes. We've got I do apologise for that. And then ribosomes. So dotted all over the rough ER, the ribosomes. They can be floating free in the cell as well. ATS ribosomes, because we're in a eukaryotic cell. Ribosome structure, well, they're sort of made of two parts. They're called two subunits. And they can either be found free floating in the cytoplasm, or they're attached to that rough ER, as I said, and they are the site of protein synthesis, and you will look at protein synthesis in a lot of detail. And then last thing we're going to talk about today is centrosomes or centrioles. Centrioles are found within a centrosome, so you find centrioles located within the centrosome, and those centrioles are like two hollow sort of cylinders, and they're arranged at right angles to each other. And what they do, as I said, they make the spindle fibers for chromosome separation in cell division. And they're made of like what are called microtubules, so like micro small tube, teeny tiny little tubes all joined together to sort of, they're like hollow cylindrical tubes, teeny tiny protein cylinders. Mainly only found in animal cells. They are in some plant cells, but they're mainly in animal cells sort of thing, and they're really, really important in the separation of chromosomes during cell division. And we'll look at cell division separately. It's a whole separate topic on its own. So, now then, here is another type of animal cell. And what I want you to do now is pause the video and have a go, see if you can identify structures A to H, and then we'll run through them. So have a go at that now. Okay, so A up here is our mitochondria. So we can see that because you can see, look, the inner membrane where it's folded, it's got these little bits coming. They're relatively big as well. A, mitochondria, site of aerobic respiration. B here, Golgi. We can tell it's a Golgi because it's got little vesicles budding off it there, so that must be Golgi. C, well, C is our nucleus here. I suppose if you'd said that was nuclear plasm, you'd have got a mark for that, but oops, apologies for that. But that's the nucleus there. Where's D? D is just pointing randomly into cells, so that must be cytoplasm, site of chemical reactions. E, plasma membrane or cell membrane on the outside, controls what comes in and goes out. F, if you look carefully on F, you can see it's covered, it's got like a dotty outline, so that must mean it's rough endoplasmic reticulum. Remember I said to you, it's not always shown connected as it should be to the nuclear envelope. But so that's rough endoplasmic reticulum, site of protein synthesis there. What am I looking for? G? G. G could be a vacuole or G could be a lysosome. Either would get you a mark. Okay. I would suggest you probably a lysosome because it's quite large, but vesicle, vacuole, lysosome all would get you a mark there. And then lastly, H is a membrane there. So H is the nuclear membrane or the nuclear envelope. Controls what's going in and out of that nucleus there. Okay, so that's one example of. So remember, in an exam, you'd never be asked to identify all those structures, 2 or 3 at the most. So what I want to do now is actually go through a couple of exam questions with you. You can see the sort of thing they're going to ask you. Okay, so first one. So here's our diagram of an animal cell right here. Okay, and then the questions are name structures X and Y. So there's X and there's Y. So you just got to name them. So what I want to do now is just look at a couple of exam questions with you. So here's our animal cell here. You can see it's a diagram. We've got our organelles that have been identified there. X, 2 X's, a Y and a Z. Now, do you remember me talking to you about the mitochondria and the fact that sometimes mitochondria are shown this way, sort of like a rod there, in terms of rod cell shape. Sometimes they're shown circular. So this is where it's been sort of cut, I would say, to you horizontally, and this is sort of vertically. And they've just chopped it in half, so you're kind of looking at the end. This has been cut horizontally down the middle, so you're looking sort of top and bottom. Yeah, so this is the same organelle, but just cut differently, as they took a picture of the cell, a slice of the cell. So name structures X and Y. So what is X and what? Just got to name them. What are they? Identify which structure is the site of aerobic respiration. So we're linking function to structure there, and describe the function of organelle Z. Okay, so have a go at that. Pause the video and have a go at that. So name X and Y. So X, this is a mitochondria here and here. Y, rough endoplasmic reticulum. You can tell it's mitochondria because you've got folded in a membrane, so you know that's a mitochondria, and you know this is a rough endoplasmic reticulum because it's covered in little bubbles. And then 2, identify which structure is the site of aerobic respiration. So aerobic respiration occurs in the mitochondria, so that would be X. And then question number 3, describe the function of organelle Z. Organelle Z is the Golgi body or the Golgi apparatus, and we can tell that because it's got these little vesicles budding off it. Okay, and the function of the Golgi is to modify molecules like protein and package them in vesicles for transport. Remember, it also makes lysosomes. Okay, another question. This one here. Describe the features shown in the diagram that allow the structure to be identified as a nucleus. So why is that a nucleus? How do you know that that is a nucleus? Okay, so pause the video and have a go at that. Okay, so the question asks us to describe the features shown in the diagram that allow the structure to be identified as a nucleus. So describing what we can see, it's a say what you see question. Well, it's got that nucleolus there in the middle, so you would get a mark for saying about the nucleolus. You can see the envelope here. Look, we can see it around the outside here. And then you can see, look, little holes or the little pores here. There's another one there. There's another one there. This is an example of electron microgrowth. This is the sort of picture that will come off that very, very high-powered microscope. So you can see that we've got the big nucleus there with a nuclear envelope, the nucleolus, the nucleoplasm, and those nuclear pores there. All those would have got a mark for that question. We can also see, if you look, these here are mitochondria. These are all mitochondria here. And this here is all endoplasmic reticulum. Okay, unfortunately my picture's a bit too blurred to see if it's rough or smooth. I think it's rough, but this is all endoplasmic reticulum that you've got here. It's attached to the nuclear envelope. Okay, so what I want you to do now is to have a go on the Moodle VLE. There's an exam question pack that you can have a go at with 5 questions. So have a go at the 5 questions, and then the answers are underneath, so you can check you can mark them effectively. And you'll bring those with you when you start college in September. Then there is also other videos for you to watch on there with exam question packs as well. So hopefully I'll see you for those. If you have questions or worries or concerns after this video, there is a playlist, so you can go and have a look at some other videos on cell structure as well. The Amoeba Sisters on there, Hank's on there as well. You can watch his videos on it. Or you will see our email addresses are on the Moodle VLE for all the teachers. I'm Saran Mullane, so if you want, you can email me with questions, but all the other teachers are there. You can drop them a line and they will get back to you. Okay, so have a go at your exam questions.